everyone. So I've got a kind of rough drawing here, and the first thing that we're going to look at is photosynthesis, the light reactions. So what I've drawn here is the thylakoid membrane, so I'm going to just abbreviate that TM. Up here we've got the stroma. Okay, and the stroma is on the outside of the thylakoid membrane, but still inside of the chloroplast. And then down here is the lumen. Okay, and that's the space on the inside of the thylakoid membrane. So remember, when we're talking about the process of photosynthesis, specifically the light reactions, we need to look at the flow of electrons, and those electrons come from water. So we have H2O, and that is going to be split into several different things here. Okay, so it's going to be split into 1/2 of an O2. Notice there's only one oxygen molecule from water, and atmospheric oxygen is usually O2, so 2 molecules of oxygen combined. So you've got your first half there. Then we also end up with 2 molecules of H+. Now remember, hydrogen has 1 proton, and to be neutral, it would also have 1 electron. But those 2 electrons are going to be used in this process. So our electrons come from this hydrogen. So we have 2 E negatives. Those are electrons. And they are going to move into the very first enzyme of the thylakoid, and that is called Photosystem 2. So we're going to call that PS2. Now it's named Photosystem 2 because it was the second of the photosystems to be discovered. All right. Now you've got your electrons in Photosystem 2, and Photosystem 2 is an enzyme that has pigments such as chlorophyll and carotenoids, and those pigments are responsible for harvesting energy from the sun. So you've got your sun's light, and it's hitting PS2, so this is light from the sun. We'll draw a little sun here so we can see that. All right, now this energy is going to be concentrated in those pigments, and it's going to be transferred to the electrons. And the purpose of this is to excite those electrons so they are going to contain energy within them. Now that energy is going to be used, and the way that that occurs is the electrons are transported, and this is an MEC, a mobile electron carrier. So the mobile electron carrier is going to transport the electrons to the next enzyme. Now the next enzyme is called a H+ or proton pump. Okay, and the whole purpose here is we are going to be taking 2 hydrogens, 2 hydrogen ions that are in the stroma, and pumping them down into the lumen. Now we already have hydrogen ions down here, so notice we're actually going up the concentration gradient. We are going from high concentration to a higher concentration. So these hydrogens up here are going to be taken away, and this is going to take energy because we are going up the concentration gradient, and that energy comes from the electrons. So we still have the electrons, but their energy is transported, the energy is used up as those hydrogens are transported across the thylakoid membrane. And we'll talk about what the purpose of those hydrogens, increasing the concentration of them, talk about what that is for. But those electrons still need to be used, so now we've got another MEC, and those electrons are going to be transported across to the next enzyme, and this one is Photosystem 1. Okay, so this was the very first of the 2 photosystems to be discovered. And like Photosystem 2, there are pigments like chlorophyll and carotenoids, so we've got our sun, and those pigments concentrate the energy, and then they transfer the energy to the electrons, so the electrons are excited again. Now that energy from the electrons is going to be used one last time, so our final mobile electron carrier is going to move the electrons over and into the final enzyme for the use of the electrons, and that is called Nadph reductase. Okay, so That's NADPH reductase. And the way that NADPH reductase works is you have a molecule of NADP plus, notice it has a positive charge. Now these two electrons are going to come in, so we've got our two E negatives. So one of those electrons is going to join up with the phosphate here, but we still have one extra electron. And we also need to deal with the H part. So you've got a hydrogen ion, and it's going to be involved in this as well. That's going to pick up the other electron, and together these molecules are going to make NADPH. Now this NADPH is super important because this is one of the final two products of the light reactions, and it is going to be used in the dark reactions. So the energy that is carried in these electrons from PS1 is stored in this NADPH. It is a high energy molecule. ATP is also required in the production of glucose during the dark reactions. So here is where this last molecule comes in, and that is ATP synthase. Okay. And ATP synthase is going to take a molecule of ADP, which is adenosine diphosphate, plus a phosphate. And notice there's going to be a bond here between the last two phosphates. And we talked about this a little bit during biological molecules. That's high energy. And then we're going to end up with ATP, which is adenosine triphosphate. Now, where does this energy come from? Well, we talked about how we were pumping all of these extra hydrogens down into the lumen, and remember that causes a concentration gradient. We have a lot of potential energy stored in the lumen now because these molecules want to go back down the concentration gradient. They want to go from here, where they are, to here in the stroma, where they are not. Okay, so we're going to go from the lumen into the stroma. Now, the only place inside of the thylakoid membrane that this can occur, where they can go back down the concentration gradient, is right here at ATP synthase. So all of the potential energy that is stored in those hydrogens as they are on the inside of the lumen is going to be captured here in this bond, and that's going to allow the third phosphate to bond there to ADP, causing us to get ATP. And now ATP is going to be used in the dark reactions or the Calvin reactions along with NADPH. So you'll notice that pretty much here all we need is water and light, and we end up with NADPH and ATP. So obviously we still have this ADP and this other phosphate and the NADP plus. All of those are recycled from the Calvin reactions once the energy that is stored in those bonds is utilized. So in the next short video, we will talk about the Calvin reactions and how these molecules are used.